And the title of your talk is A Virus-Based Nanoparticle Delivery System.
Good afternoon. I'm Jennifer Kefauver, and I'd like to talk to you about the construction of a virus-based nanoparticle delivery system. As anyone who has experienced or witnessed the devastating side effects of radiation and chemotherapy knows, there's a problem of specificity in the current cancer treatment paradigm. Although we know many ways to interrupt oncogenic processes at the molecular level in vitro, we are limited by our inability to target these potential therapies specifically to cancer cells in the body. The Spano Lab has taken the approach of harnessing the natural nanoscale delivery function of a viral system to carry cancer drugs directly to the source of the problem. How do we plan to do this? We want to build the adaptosome. We envision the adaptosome as a cargo delivery vehicle whose backbone is the OR5 prohead, a self-assembling hollow protein shell. The prohead would encapsulate small cargo, for example, this 5 nanometer gold particle. Dual function adapter peptides would attach to the outside of the prohead via non-covalent interactions with the prohead surface via this red prohead attachment sequence, and then carry the whole complex across cell membranes with this green cell penetration sequence. The idea is to put all of these components together in one reaction to form a vector that can be adapted to each patient's needs. Let's look at each of these components more carefully. The OR5 viral prohead is composed of the major capsid protein from a defective virus derived from Rhodobacter capsulatus. It is considered defective because it does not package its own DNA to propagate. Instead, it randomly encapsulates short sequences of the Rhodobacter genome and carries it to nearby cells. This system is optimal for our purpose because it is essentially a blank slate that will accept any cargo of the appropriate size and charge. It also has another exciting feature, that is, it self-assembles in the presence of calcium ions. Why do we believe this happens? This chromatogram of a size exclusion experiment shows that OR5 with calcium added, the far left piece, is part of the excluded volume and thus is larger than the OR5 protein with no calcium. This effect can also be seen in Kumacy-stained native agarose gels. In lane 1, the pentameric subunits of OR5 with no calcium added travel slower into the gel than the formed prohead in lane 2. So we know that calcium causes a change in size, but how do we know that a virus-like particle is being formed? These images, acquired by 2 separate methods, show that OR5 is a spherical shape with a diameter of about 30 nanometers. The image on the left was acquired by transmission electron microscopy and compares the small OR5 viruses, viral proheads, to the 100 nanometer herpes simplex virus in the center of the image. The image on the right was acquired by scanning transmission electron microscopy with OR5 proheads in a cryo state, comparing them to the Paco mosaic virus, the filamentous virus in the bottom right-hand corner, who has a diameter of about 20 nanometers. So we have a self-assembling protein shell, but what can it do? I have been able to show that OR5 can encapsulate 5 nanometer gold particles. Gold nanoparticles are an interesting cargo because when they're hit with a relatively low intensity of laser light, they begin to vibrate and create local heating, which can be used to thermally ablate cancer cells. This urinal acetate stained TEM image shows the 5 nanometer gold particles, the dark spots of high electron density, in the center of OR5 capsids. These were prepared by briefly incubating OR5 protein with 5 nanometer gold particles at approximately a 2-to-1 prohead to gold ratio, followed by the addition of calcium chloride. I have seen an encapsulation rate of about 23% of proheads being filled in all the images that use this ratio. This can be increased by a certain degree by changing the gold to protein ratio, but at a certain limit of gold concentration, prohead formation kinetics are affected and an aggregate product is formed. Beyond that, 10 and 15 nanometer gold particles have also been incorporated. But how do I know that the gold particles are actually inside the virus, inside the prohead? First of all, I never see the gold particles anywhere except for the center of the protein shell. If they were stuck to the outside, then we might see gold particles stuck at any location along the edge of the prohead or inside the prohead. Secondly, if I change the order of addition and add the calcium first, allowing proheads to form before adding gold, the gold aggregates in the presence of calcium and does not associate with the protein. In lane A are proheads alone, so you can see that the addition of gold does not change the rate of travel of the proheads through the gel. In lane B, gold is added before calcium, and you can see in the top non-stained image that it forms a band similar in the same location as the proheads, and a hole was poked in the gel before staining to show that the gold and the protein are co-localized. In lane C, proheads were formed first by adding calcium first, and the gold in these does not travel into the gel, but instead sticks in the well in an aggregated form since the calcium destabilizes it. From this experiment, I learned that not only is the prohead encapsulating the gold particle, but it is also protecting the gold from a calcium-induced aggregation effect. I got excited about this because it means that the prohead can function not only to carry cargo into cells, but also to protect it. To further examine the protective effects of the OR5 prohead, I exposed bare gold nanoparticles and gold encapsulated by OR5 proheads to increasing concentrations of sodium chloride. The absorbance of intact gold nanoparticles is 5 nanometer gold particles is 515 nanometers. As the salt concentration increases, the gold becomes completely aggregated and its absorbance shifts to higher wavelengths, to longer wavelengths. However, the gold inside proheads does not experience this effect and absorbs at 520 through the highest salt concentration. So now we know that OR5 is an excellent candidate for carrying cargo around in vivo, but how will it deliver its cargo on target? The Spanner Lab has chosen to use the approach of non-covalently attached adapter peptides. First, I needed to find a peptide that would bind non-covalently to the surface of the OR5 prohead. To do this, I used a method called phage display, developed in the 80s by the George Smith lab. The gene that codes for the low-copy terminal coat protein of the M13 phage is known, so libraries of phage with different amino acid sequences in that location can be engineered. To find phage that bind to the prohead, I mixed the phage and the OR5 proheads in solution and incubated them for an hour, then washed away the non-binding phage. The binding phage were eluted at pH 2.2, and the gene for the 7-amino acid N-terminus of the coat protein was sequenced. That peptide sequence can then be linked to a cell penetration or attachment sequence. I found that there was a consensus among the binding phage, a sequence that we call GTA1. It is highly hydrophobic with 3 tryptophans, and it has an alanine, a charged residue, in the center. This sequence was then synthesized with a 2-glycine spacer and a cysteine at the C-terminus to allow it to be linked to cell-targeting peptides by a disulfide bridge. The next question to ask was what cell-penetrating peptide should be used to carry the prohead across the cell membranes. We decided to use a general cell-penetrating peptide at first. PEP1 peptide is available commercially as Chariot and was chosen because the cargo attachment portion of this peptide looks remarkably similar to the GTA1 sequence. HYBE2 is the result of the successful conjugation of GTA1 with TAP peptide derived from HIV1. It's commercially available. This was accomplished by a disulfide bridge interaction followed by a purification by HPLC. Let's first look at the results of cell penetration by OR5 with Chariot. Chariot was incubated with Alexaflor 594 labeled pentamers and labeled OR5 proheads at a ratio of 1 to 130 protein to peptide. In the image on the left, pentamers were carried in and a cytoplasmic labeling of the cells by fluorescent pentamers is visible. However, in the right image, proheads only appear to label the surfaces of cells, presumably because the proheads are too large to be carried in by Chariot peptide. However, this result was not unexpected because Chariot peptide was not shown in the literature to carry proteins on the megadalton scale into cells. After this, my attention was shifted to HYBE2 peptide because it utilizes the GTA1 prohead attachment sequence, and thus it could be varied by attachment to tumor-homing peptides instead of a general cell-penetrating sequence. But first we need to know, does the HIVE-2 peptide bind to OR5 proheads? To test this, my mentor and I designed an experiment using sucrose density gradient centrifugation. When centrifuged at 15,000 RPM for 6 hours, the fluorescein-labeled HIVE-2 peptide does not travel into the gradient alone. However, when OR5 proheads are added, it travels into the gradient into 2 tight bands, presumably 2 different size complexes. This preliminary result indicated that HIVE-2 peptide binds to OR5 proheads in a complex way. The binding will be further investigated with a concentration series of sodium chloride to determine if the interaction between HIVE-2 and OR5 proheads is a charge-dependent or hydrophobicity-dependent interaction. So far the things that I have done are demonstrated the self-assembly of OR5 proheads. I have shown that proheads are capable of encapsulating 5 nanometer gold particles, and the non-covalent prohead attachment sequence has been revealed by phage display. One has been hybridized with cell-penetrating TAT peptide and shown to bind in a complicated way to OR5 proheads. What's left to do is to establish cellular uptake of OR5 proheads with HIVE-2 peptide and to link GTA1 with tumor-homing or transcytotic peptides to carry them to specific cells. And we need to demonstrate that in culture. Eventually, we also want to diversify cargo and include different nanoparticle structures or gene products. I'd like to thank my mentor, Dr. Anthony Spano, and my lab partner, Hilary Sloan. I'd also like to thank the Keck Center and the Advanced Microscopy Facility for training me to use their instruments, and the Harrison Institute for funding. Are there any questions? Yeah.
I may have missed this. Was GTA1 your only hit in the phage display screen?
There was another peptide that was similar to GTA1 that had a tyrosine replacing the tryptophan in one location and a couple of other similar changes like that. But we got the GTA1 more often, and we thought they were similar enough that that one would be successful.
So I have a very general question. What's this term adaptosome?
Adaptosome, it's used because we'd like the entire complex to be adaptable to each patient or to several different types of cancer cells. It's as general as you can get. You could also change the peptides to have a transcytotic property so they can be carried across the blood-brain barrier. And you can also mix different types of peptides so that you could do both. You could carry it across the blood-brain barrier and then specifically to glial tumor cells.
I guess I'm wondering, does it have applications outside of what you're doing?
Probably. It could be used for anything where you want to target cargo to a specific place in vivo. Any other questions?
Okay, thank you, Jen.